Hi, welcome back. We're in the playlist on the pentose phosphate pathway, and what I want to do here is I want to first look at that reaction that I talked about, and I'm going to draw ribose 5-phosphate, and I'm specifically going to draw it in its cyclic form. In the last video, we actually looked at ribose 5-phosphate in its linear straight chain form, but what I'm going to do here is I'm going to draw it specifically in its cyclic form. And what I have here is I have the cyclic form of ribose 5-phosphate, and specifically this is the what? This hydroxyl group's going down, so that means it has to be the alpha. This is the alpha configuration of the anomeric carbon. And ribose 5-phosphate can get consumed by an enzyme called phosphoribosyl pyrophosphate synthetase. And usually we abbreviate it PRPP synthetase. And what this enzyme is going to do is it's going to burn an ATP, and it's going to attach a pyrophosphate linkage ultimately to the anomeric hydroxyl group. So you're going to put in an ATP, and you're going to get out adenylate. You're going to get out adenylate, and so you're going to generate something that looks like this. You're going to generate something that looks like this. So I still have my phosphate on the 5 position, but I have this. And so this right here, this group essentially is my pyrophosphate linkage. So this molecule is called phosphoribosyl pyrophosphate, or more specifically, 5-phosphoribosyl 1-pyrophosphate. And this molecule right here is used in the biosynthesis of it's used in the biosynthesis of all the nucleic acids, all of the whether it's AMP, GMP, all the pyrimidines. It's used in all of them, and several amino acids. It's also used in the synthesis of them. So what can happen is the ribose 5-phosphate can essentially be pulled out of the pentose phosphate pathway, and it can be consumed by this enzyme and used for biosynthesis. And what you're going to find is that several of these molecules can actually be pulled out and used in other biosyntheses. And actually what you'll also find is that if we produce one of these molecules in another, whether it's a catabolism or another biosynthesis, we can put them into the pentose phosphate pathway. Okay. So what I want to do in this video is look at the rest of the pentose phosphate pathway, and specifically it's the non-oxidative. And the reason it's called non-oxidative is in each particular reaction there's no net change in oxidation state. So we're not dehydrogenating, we're not doing anything with NADPH, we're not doing anything with NADH, and essentially there's no net change in oxidation state. So what I want to do is I'm going to come down here, and I want to show you that ribose 5-phosphate, and what I'm going to do in this video is I'm really just going to abbreviate the structures by their names. You can certainly look in a textbook to figure out the structures, but for the purposes of right now, we're going to really just look at the names, not the structures. Okay, so the first reaction is catalyzed by an enzyme called transketolase. Called transketolase, and transketolase is an enzyme that essentially is going to move the ketone of the keto sugar onto the aldo sugar, right? It's essentially that's a transketolase. It's going to move the keto group. So what it's going to do is it's going to specifically transfer 2 carbons. So transketolases, so this is catalyzed by transketolase, and transketolases specifically transfer 2 carbons. Okay, so there are 2 carbon transfer molecules, and I think it's helpful to actually keep track of where the carbons are. Okay, so ribose 5-phosphate is 5 carbons, and I'm going to show the carbon number in blue. And essentially what you're going to put into this is another 5-carbon sugar, and it's a 5-carbon keto sugar, and it is Xylose 5-phosphate. What you'll find is that all of these sugars that we're putting in are phosphosugars. So xylose is a 5-carbon sugar, So this is a 5-carbon sugar. And so logically, if we're transferring 2 carbons, one of these sugars is going to go down by 2 carbons, and the other one's going to go up by 2 carbons, right? So the goal is just to figure out which one goes up by 2 carbons. Well, if we're transferring the keto group to the aldose sugar, right, it would make sense that the aldose sugar would go up by 2 carbons, right? And that's exactly what's going to happen, right? So the Xylose is essentially going to transfer its keto group. Essentially, it's really just a net change. The mechanism is a little bit more complex. But essentially, the ribose 5-phosphate is going to receive the 2 carbons. And it's going to generate another keto sugar. And that keto sugar is called acetoheptulose 7-phosphate. And of course, this sugar has 7 carbons. And so that means that the Xylose 5-phosphate lost 2 carbons, and so we're going to generate an aldose sugar, and specifically it's glyceraldehyde 3-phosphate. And that, of course, is part of glycolysis, although most likely this G3P is not going to go into glycolysis. It's actually going to be consumed by the next enzyme. And the next enzyme is called a transaldolase. It is called a transaldolase, and in fact what it's going to do, and let me go ahead and draw the reaction. It is called a transaldolase. It's going to take the acetoheptulose 7-phosphate and the G3P, but the thing you have to understand, let me write this, it is a transaldolase. But what this enzyme does is instead of transferring 2 carbons, it's a 3-carbon transfer molecule. It's a 3-carbon transfer molecule. And so what's going to happen is it's going to, the net effect is it's going to transfer 3 carbons between these molecules. And so you can expect if it's transferring 3 carbons, one of them is going to go down by 3 carbons, and the other one's going to go up. And you can sort of reason through this, right? Glyceraldehyde 3-phosphate is 3 carbons, right? So it's not going to be the one that's going down by 3 carbons, right? It's going to have the acetoheptulose 7-phosphate is going to go down by 3 carbons. And so if acetoheptulose 7-phosphate goes down by 3 carbons, what's going to happen? Well, it's going to generate a 4-carbon sugar, right? It's going to generate a 4-carbon sugar. And that 4-carbon sugar is called erythrose 4-phosphate, right? Erythrose 4-phosphate. And so that means that the G3P, the glyceraldehyde 3-phosphate, has to go up. It has to go up by 3 carbons. So it's going to be a 6-carbon molecule, and it's going to generate fructose 6-phosphate. And the good thing about the pentose phosphate pathway is that we actually generate intermediates in glycolysis. And so those particular molecules can actually be shunted back into glycolysis or gluconeogenesis depending on which direction you're going. So what I'll go ahead and do is I'll show you this, that fructose 6-phosphate actually leaves and gets shunted back into glycolysis or gluconeogenesis depending on which direction we're running, right? So we've essentially lost this fructose 6-phosphate. It's no longer part of the non-oxidative pentose phosphate pathway. However, erythrose 4-phosphate is, right? Erythrose 4-phosphate is still part of the pentose phosphate pathway. Okay. Now, the erythrose 4-phosphate, remember, is an aldose sugar, and just to illustrate this, it's 4 carbons, right? And it's going to get reacted with another enzyme, and this is the same enzyme that we started with, and it is transketolase. And so it's going to have a net transfer of 2 carbons, right? So you can imagine that erythrose 4-phosphate, it's probably not going to go down by 2 carbons, it's going to go up by 2 carbons. And it's going to get the carbons ultimately from, Xylose 5-phosphate. And that has 5 carbons. And the way I remember this is the fact that you start and end with transketolase, and you also start and end with Xylose 5-phosphate, right? The initial substrate, one of them, was Xylose 5-phosphate. So not only do you start and end with transketolase, you start and end with Xylose 5-phosphate. And so you can imagine that the carbons, those 2 carbons, are probably going to come from Xylose 5-phosphate, And so that means that erythrose 4-phosphate is going to go up by 2 carbons. And it's going to generate fructose 6-phosphate. And of course, just like the last fructose 6-phosphate, that can ultimately go into glycolysis or gluconeogenesis, depending on which way we're running, right? Okay, and so if the erythrose 4-phosphate goes up by 2 carbons, that means xylose 5-phosphate has to go down by 2 carbons, right? And so we're going to generate another glyceraldehyde 3-phosphate, right? And then that glyceraldehyde 3-phosphate can be shunted into glycolysis, right? And it's going to be consumed by whatever enzyme there is, depending on which direction we're running. So what do we see? Well, the oxidative pentose phosphate pathway, which was the previous video, what we saw is that we generated 2 NADPHs per entry into the pathway, right? And the NADPHs, by the way, I'll mention, are absolutely essential to life. You cannot live without NADPH. And one of the mistakes that people make is a lot of times in general biology classes, you talk about NADPH only with respect to plants. And I was the same way for a while. I thought that NADPH was just in plants. But no, NADPH is used in biosynthesis reactions of all types. And what you would find is that if you went into more advanced biochemistry, pretty much every biosynthetic pathway uses an NADPH or uses an NADP, depending. Okay? But the non-oxidative pathway, what it's doing is it's generating substrates for other biosynthesis reactions, and we'll actually look at an example of these in a few minutes. Okay, but what I want to do now is I just want to kind of regroup. The purpose of the non-oxidative pathway is to, first of all, generate things that can go back into the glycolytic pathway, and those things would be fructose 6-phosphate and glyceraldehyde 3-phosphate. And the other purpose of it is to generate substrates that we can siphon out and use in other biosyntheses. And certainly we've already looked at one of those. It was PRPP synthetase to generate phosphoribosyl pyrophosphate. And of course that can go into biosynthesis of nucleic acids and other things like histidine biosynthesis. Okay, now what I want to do is I want to look at some of the other things we can do, right? Now what I want to do is I want to draw the structure of xylose 5-phosphate. So here's going to be xylose 5-phosphate, and recall that xylose 5-phosphate is simply an epimer. It is an epimer of ribulose 5-phosphate, right? It's an epimer, okay? So what that means is that it differs at the stereochemistry about 1 carbon, and it's this carbon. Recall that in ribulose 5-phosphate it's pointing the other direction. So here is, let me write this. This is xylose 5-phosphate, and there's an enzyme that essentially removes this hydroxyl group right here. So it essentially removes this hydroxyl group, and the enzyme is called 1-deoxy xylose 5-phosphate synthase. And essentially what it generates is something that looks like this. It generates this. And this is essentially 1-deoxy xylose 5-phosphate. So we essentially remove that hydroxyl group as water, and we generate 1-deoxy xylose 5-phosphate. Well, it turns out that this particular molecule can actually go into other biosyntheses. So what we can do is we can take xylose 5-phosphate out of the pentose phosphate pathway when we generate it, and we can use it in other biosyntheses. And one of the biosyntheses that it's used for is the biosynthesis of thiamine pyrophosphate. And it turns out that thiamine pyrophosphate is it's a cofactor for many enzymes, right? And if you recall, it's a cofactor. It's a cofactor for the pyruvate dehydrogenase complex, right? It's also a cofactor for another enzyme called acetolactate synthase, which is involved in branched chain amino acid biosynthesis. Another example of a place we can use this is pyridoxal phosphate. is pyridoxal phosphate biosynthesis. Now, what I want to be perfectly clear about is none of these biosyntheses, at least the synthesis of thiamine pyrophosphate and pyridoxal phosphate, these are not done in humans, but they're done in bacteria. But essentially, they use the xylose 5-phosphate from the pentose phosphate pathway. And pyridoxal phosphate is used in a lot of reactions. One of the most important reactions it's used for is transaminases. And if you take a class that talks about amino acid catabolism and amino acid biosynthesis, and even nucleic acid biosynthesis, you'll see a lot of transaminases, and what they'll typically do is they'll put in parentheses above the enzyme name, they'll put PLP, and that just stands for pyridoxal phosphate. So anyways, xylose 5-phosphate can be siphoned off of the pentose phosphate pathway and used for other biosynthesis. Let's look at another example. Another example would be erythrose 4-phosphate. So erythrose 4-phosphate is essentially an aldose sugar, and it's 4 carbons, right? And so here's what it looks like. This is erythrose 4-phosphate. And erythrose 4-phosphate can be used in many syntheses, but the most notable one, and by the way, this is another reaction that does, as far as I know, is not used in humans, but it's involved in the synthesis of charismate. And then charismate can be then used to form the aromatic amino acids, and those, of course, would include tryptophan, tyrosine, and phenylalanine. So, and the initial reaction that uses erythrose 4-phosphate is 2-amino-3-deoxy D-arabinoheptylosonate 7-phosphate synthase. So essentially, this is another example. We can take something out of the pentose phosphate shunt, and we can use it for biosynthesis. And in general, that is the main function. That is the main function of the pentose phosphate pathway. At this point in my career, I don't really know of any reactions that use cetoheptylose 7-phosphate, but the main things that we're going to take out and use for biosynthesis are xylose 5-phosphate and erythrose 4-phosphate. And of course, like I mentioned, a lot of these reactions don't occur in humans, but I think the big point from this video, besides the actual reaction scheme, is to understand that the purpose of the non-oxidative phase is to create substrates for other biosyntheses. And obviously those molecules can be used as cofactors and things like that. And certainly for, as we saw, biosynthesis of the aromatic amino acids. Okay, and remember that the purpose of the oxidative pathway is to produce NADPH. And remember, you cannot live without NADPH. You have to produce it. And this pathway, at least the oxidative phase, is the most important source of the NADPH. So I hope this video helped you get an intuitive sense of what the pentose phosphate pathway is and what it's for. In future videos, we may look at other aspects of it. But for now, the most important thing to understand is the actual pathway, and then, of course, the applications of it. See you soon.